Hello everyone, today I'm going to introduce some work I've been doing on the genus Packera within Asteraceae. So to start off, Packera as a genus is endemic to North America and is reported to contain about 64 species and varieties, although the number is potentially as large as 88 or higher. It was previously included in the genus Senecio as the informal group known as the Orioid Senecios, but was eventually determined to be a separate genus in 1981 based off chromosome numbers, various morphological differences, and molecular phylogeny data. Ecologically, Packera is adapted to a variety of habitats, so some species are abundant and widely distributed, such as Packera, which is a widespread species found throughout most of the West Coast, while others are narrow endemics that are restricted to specialized or isolated habitats, such as Packera franciscana, which is a threatened species found only in Arizona's San Francisco peaks. Taxonomy remains complex within Packera due to the species' ability to easily hybridize and polyploidy being common throughout the genus, with roughly 40% of taxa being polyploid. And a great example of a taxonomically complex species is Packera palpercula, which is a widespread species that is ecologically and morphologically variable along its range, and it is believed that most of this variation is due to polyploidy, hybridization, and introgression. So this complexity has made it difficult to understand the evolutionary relationships within Packera, and other researchers have tried by generating Packera-specific phylogenies to gain a better understanding, such as this phylogeny generated by Bain and Golden in 2000. However, the phylogenies have many unresolved areas, which are represented by these comb-like structures called polytomies, and can potentially be explained by the use of fewer phylogenetically informative sites, lower species sampling, and potential hybridization or articulation between species. Additionally, this phylogeny only accounted for nuclear genetic diversity, excluded plastid variation, which is necessary since further research into Packera has shown there's a high amount of plastid diversity present within the genus. Therefore, I plan on generating a robust nuclear and chloroplast phylogeny of Packera, as well as estimate its age. And by doing so, I hope to address, is Packera currently circumscribed monophyletic? Do the resulting species relationships agree with what we previously knew about Packera's history? Do plastid and nuclear phylogeny share the same topology? And then what is the estimated age of Packera as a genus along with its members? So far, I have sequenced 83 of a targeted 93 Packera taxa, giving me an 89% coverage of the genus. I've also sequenced 83 outgroup taxa, giving me a total of 106 individuals, and about an 88% coverage of all targeted taxa. Given limited time and resources, I'll probably stick with this 88%. But then using the 106 individuals, I then performed HiveSeq methods that target 1,061 conserved orthologous set loci to get nuclear sequences needed to build the phylogenies. And then I did not target chloroplast sequences, but instead used the software Botany, which extracts off-target chloroplast sequences that can then be used to generate plastid phylogenies. And so once I obtained both nuclear and chloroplast sequences, I used multiple pipelines to find the best fitting tree. So for the nuclear sequences, I used the Piper and Lucci pipelines for ortholog detection. And then output of those pipelines were used as input for pseudocoalescent concatenated analyses. And then the trees were inferred using a maximum likelihood approach using the software RaxML. For the chloroplast phylogeny, when sequences were obtained from Bowtie, I then ran them through the online program GSeq, which annotates genes within the sequences. I then used Genius to extract those annotations to then be able to use as input for pseudocoalescent concatenated analyses, which were then also inferred using RaxML. For the sake of time, I'm only going to be showing the nuclear Piper pseudocoalescent RaxML tree, which is the tree that I found to be the best fitting. But if you have any questions about any of the other trees, I'd be more than happy to discuss them after this talk. And so here's the resulting tree of 106 taxa. It's comprised of 1,049 loci and is almost 531,000 bases long. I separated the tree based on outgroups and ingroups. And so ingroup taxa, or the taxa of interest, and in this case Packera, have white branches, while the outgroups are colored either purple, green, blue, or teal. The teal outgroup, which is the where the tree is rooted, which is on the bottom of the tree, is comprised of 4 taxa within the tribe Anthemidae. And I included these outgroups because there's a fossil pollen for Artemisia that'll be used when dating the tree. And then the remaining outgroups are other taxa that are within or sister to tribe Senecioneae. When trying to understand the tree, I removed all branch support numbers, but instead labeled them with icons indicating whether they had a local posterior probability value, or LPP, of 1 as red stars, or an LPP value of 0.95 or higher with a blue triangle. LPP values range from 0 to 1, 1 indicating the highest support, and they indicate the probability that the branch is the true branch given the set of gene trees. And when running Astral, it's suggested to use LPP instead of bootstrapping to find support. Just for some stats, 54 of the 105 nodes have an LPP value of 0.95 or greater, and then 44 of those have an LPP of 1. And then additionally, whenever you run Astral, you get something called the normalized quartet score that tells you how many of the gene trees match the final species tree. And in this case, we got a value of 0.493, indicating that 49% of the gene trees match the final species tree. And then given how large this tree is and the species name are quite hard to see, I include a QR code on the top right that will link you to this tree with all LPP values in case you have any species specific or nodal support questions. But using this phylogeny, we then wanted to see if there are any trends and traits to the relationships that this phylogeny proposed, and if we can relate that to what we previously have known about Packera's history. So in the next few slides, I'm going to show you a simplified version of this tree along with some things that I want to highlight, such as we wanted to see if traits such as morphology, base chromosome number, or ploidy or geography show any indication of evolutionary relationships. And so when looking at these, we're trying to see if there's any trends within these traits. And so for the first one, morphology, it does not seem to be showing any traits. So I have highlighted 3 sets of individuals who have been reported as being morphologically similar in the field or from herbarium sheets. And as you can see in this phylogeny, although these individuals are known to be morphologically very similar, they appear to be quite distant genetically. Therefore, morphology is not really looking like a very strong indicator of evolutionary relationships. When we look at Packera's base chromosome counts, which are mainly 22 or 23, with some individuals having 20, we see that there does not seem to be any trends. So these are Packera species that have a base chromosome number of 23, 22, and even the rare 20. So there doesn't seem to be very many trends with base chromosome number either. Additionally, when we map ploidy onto the tree, we see that polyploidy does not seem to be a good indicator. And so here I've highlighted all species that have reports of polyploid counts. So that could be from tetraploid to octoploid and beyond. And so again, ploidy level also does not seem to be an indicator for evolutionary relationships. What seems to generally be a better indicator is geography. And so here I have mapped general distribution of Packera species along the phylogeny and color-coded them according to general geographic areas where the species occur. And so here we can see trends such as West Coast or Californian species are all within one clade, which we can see here in the blue. And that trend also continues with East Coast species highlighted pink and Arctic alpine species highlighted red. But we start to lose that trend when we map Southern US and Mexican species or Central US species, which seem to be distributed throughout the phylogeny. And so though this trend is not perfect, there does seem to be some clade-specific trends of geography, which is quite interesting, though not surprising, as other researchers into Packera had previously predicted that geography would be important in Packera speciation events. So far we're mostly seeing that geography generally seems to be a better indicator of evolutionary relationships, at least more so than morphology, base chromosome number, or ploidy level. So just some other general things with the phylogeny. When we look at Senecioneae as a whole, the phylogeny also shows some subtribal separation between Senecioneae and Tussilaginae, which is great and what you'd like to see. Additionally, when we also see that some species such as Tephroceras and Doronicum are placing outside and more or less sister to the Senecioneae clade. These species relationships are not surprising since both taxa's placements with or within Senecioneae continuously change. That usually depends on the different taxa included in the phylogeny or the sequence type. But previous researchers had suggested that these taxa actually be elevated to subtribal status, though nothing has become official with that. But this phylogeny provides evidence for their subtribal status's validity. So though more research is necessary. One of the most interesting things I found with this phylogeny is that some Packera species are actually grouping within the outgroups. And so, therefore indicating that Packera is not monophyletic. Something interesting about these species, though, is that most of them were previously predicted to actually be misplaced into Packera. And so other researchers had found that they are morphologically different, they have a different chromosome count, or their pollen type is different. And they believed that they should not actually be in Packera, but potentially be placed back into Senecio. And so this phylogeny is providing further evidence that these species should actually be separated from Packera. And given our phylogeny, it's in a Senecio clade, indicating that yes, it maybe should be put back into Senecio. Something that is interesting, though, is that one of the species within the group, Packera Moorani, was always known to be a good Packera species, and so that's quite a surprise to see it in with the outgroups. So it potentially needs a taxonomic revision, though that goes beyond the scope of this study. And so bringing it back to the research questions, there's really only two that I can address today. So one is Packera monophyletic. Well, as I just showed you, no, although the Packera species that are in the outgroup were predicted to be actually within the genus Senecio, it's still taxonomically still not monophyletic. And then do the resulting species relationship of phylogeny agree with what we previously knew? Yes and no. We have seen that some trends hold and then also some don't. So it's kind of a mixed bag with that. And so just some final remarks. So far, we built the most extensive Packera phylogeny to date. When looking at traits, geography seems to have the most influence when explaining species relationship, at least more so than morphology, base chromosome number, or ploidy level. Some species relationships are surprising and unexplained. Not everything was shown today, but we believe that the potential hybridization, integration, or reticulation may account for some of the species placements that we're seeing in the phylogeny and will need to be investigated further. But with that, I would like to say thank you for listening, and I will take any questions.